To make new humans, we have to get a sperm and an egg together. But how does that sperm find its way to the egg? It has to swim quite far using a flagellum. Once it's at the egg, they have to start dividing millions of times to make a new human. How does this miracle of life happen? Surprisingly, the answer is centrioles. First, to understand what a centriole is and what it does, we first need to understand the parts of a cell. Cells are the basic building blocks of life. All living things are made of cells. Within cells, there are tiny working parts called organelles. Organelles each have a different job in the cell. A centriole is an organelle that helps cells divide or make copies of themselves. Centrioles are only found in animal cells. All centrioles are made of protein strands called microtubules. Centrioles are made of 9 triplets of microtubules arranged in a cylinder. Think of each microtubule like a plastic PVC pipe. The pipes are arranged with 3 put together in a triplet, then 9 of those triplets are glued together to form an even bigger pipe. When 2 centrioles come together, they usually make a right angle. Centrioles help form 3 important structures for cells: the centrosome, cilia, and flagella. Let's start with centrosome centrioles. These words sound pretty similar, huh? To really understand the difference, we need to understand cell division, or mitosis. Mitosis is a process where cells divide and make 2 new identical cells. Before mitosis can start, the cells need to make 2 copies of their DNA. Each one will go to a new cell, so that the new cells have the exact correct amount of DNA. The centrosome and centrioles are crucial for this process. During cell division, 2 centrioles come together with some other special proteins and form the centrosome. This is an image of a centrosome, made of 2 centrioles and microtubules. Let's think of an analogy to keep these 2 vocabulary words straight before we move on. The centrosome can be thought of like a shipping box being delivered to your door. The shipping box is like the centrosome. Inside the box are 2 presents, the centrioles. Each present, or centriole, is wrapped separately. But the shipping box is important too. It's made of its own cardboard and tape and brings the presents together safely to your house where you can enjoy them. The centrosome encases the centrioles with some extra proteins, just like the shipping box encases your presents. Now, during cell division, there are 2 centrosomes, each with 2 centrioles. Here's what happens. 1. The centrosomes move to opposite ends of the cell, where they make even more microtubules. 2. The microtubules grow towards the center of the cell and attach to the duplicated DNA. 3. The centrosomes pull the DNA copies apart, giving each new cell just the right amount of DNA. Centrioles are also used in the cell structures cilia and flagella. Cilia are lots of small projections on cells, like little hairs that whip back and forth to move the cell, or move material over or around the cell. Cells in the lungs, for example, use cilia to move mucus out of the lungs. Flagella are similar to cilia, except that a cell can only have one long projection, kind of like a tail. It is used for movement of the cell only. The tail on a sperm is a flagellum, twirling to propel the sperm forward. Cilia and flagella have a similar centriole structure. There is a difference between centriole structure inside and outside the cell. Cilia have a specific centriole on the inside of the cell anchoring it to that cell, called a basal body. It has the same pattern as centrosome centrioles, which have 9 sets of microtubule triplets. Centrioles on the outside of the cell, making up the projected cilia, have 9 sets of microtubule doublets instead of triplets. They also have an additional doublet in the center, organizing the centriole differently. The purpose of cilia and flagella is to move, so the outer centrioles act like a motor. Along with other special proteins, the centrioles rotate, causing a beating motion in cilia and an undulating motion in flagella that propel them through the environment. Let's review. Centrioles are an organelle inside animal cells that are made of microtubules and are involved in cilia, flagella, and cell division. Centrosomes are made of a pair of centrioles and other proteins. The centrosomes are important for cell division and produce microtubules that separate DNA into 2 new identical cells. Cilia and flagella also use centrioles, but for the purpose of cell movement and to move liquids around cells. Inside the cilia or flagella, there are doublets of microtubules surrounding another doublet in the center, making them structurally different from centrosome microtubules.